Okay, thank you for the introduction and the invitation to speak. So indeed I was asked to talk about genetics and epigenetics of the stress response. And I have to say that when I started reading into it a bit more than I usually do, I got really scared because the amount of data that's available is really huge. And even if I would grasp all of it, there wouldn't be time to talk about all of this. So I decided to stick to the HPA axis. And even then, first thing you do is, of course, look at patient studies, or I'm interested in IBS, like was just introduced. And you look at IBS versus control studies, then the problem is there's not much consensus on the data. So most of what I'm going to talk about today will be on the animal models. And most is animal models of early life stress exposure. And I think yesterday already you heard a lot about these models and why people use these models, us included, in the lab in Amsterdam. They are early life exposure of adverse early life events are associated with IBS and with a lot of other diseases, but also with IBS. And in this paper by Lin Chang's group, you see that it's also associated with enhanced HPA axis responsiveness, not only in IBS patients, by the way. In this recent review, there's a lot of these animal models are gathered, and what you will see from that is that mostly what people use is the maternal separation model. So a lot of the work I'm going to show is also on this model, but also on another model, you will see. With some variations, most of these models will use maternal separation from day 2 to 14, so 12 days and then 3 hours a day. And what they will find is that if you compare baseline conditions, so control animal and a maternal separated animal, there's not much difference. But if you give them a stressor, you will get increased HPA active responsiveness, and that will be in all 3 levels, so CRH, ACTH, and corticosteroid. This is actually the first paper that was published with the maternal separation model, and it's by 2 people we talk a lot about in this presentation. That's Paul Plotsky and Michael Meany. And they actually, before that, they used another model where they had short handling periods of rat pups, or rat pups, yes, and a short time separation. They call it H in the next slide. It's 15 minutes, and what they saw is that there was increased mother-pup interactions in that model. And this increased tactile stimulation would lead, if you compare to normal control animals, to a lower HPA axis responsiveness. So they took along that model as a control here. Non-handled, H, and also maternal separation. So that's the 12 days in a row, 3 hours per day. And in those animals, you find an increased HPA axis response. And that's shown here in this very early paper, where they look at mRNA CRF in the hypothalamus, so in the top of the HPA axis. And you clearly see that the maternal separation at the highest levels. Now actually, so the first author in this paper was Paul Plotsky, and Paul Plotsky actually helped out in Emran Meyer's lab to introduce this model. I think Emran was the first to use it in IBS-related research. So he compared non-handled animals with maternal separation animals, and then at the adult age, he would give both groups a acute stress, a water avoidance. 1 hour in this cubicle on a pedestal, and it's considered a mild psychological stressor. And before and after, they would do colorectal distensions and measure the visceral motor response, and that's what you see in the lower picture. And if you compare before and after water avoidance, the non-handled with the maternal separated animals, you clearly see that in maternal separation you get an increased response to distension. In the same lab, so 2 years after that, Taché and Million, they show that this is really an HPA axis dependent visceral hypersensitivity. If they block central CRF receptors, CRF1 in this case, then you see that the increased response is no longer there. And this was pre-treatment. I'm going to take a small step back. This was the first paper on the maternal separation model, Plotsky and Meany. What they did, they published a couple of papers with this, and then they switched actually to another model. And a lot of data that we use now as, or know as common knowledge were generated from that other model. And I'm going to talk about it a bit. The reason that they switched was, okay, they found this change mother-pup interactions, and they saw a nice phenotype, and then they thought, okay, this is experimental induced, but what does it mean in real life? Would this be a natural phenomenon? So what they did, they took a big group of mothers, dams, called, but mothers that were just delivered, and they looked at the phenotype. What was the behavior of these mothers towards the pups? And then you see a gradual scale, and there's a what they looked at was licking and grooming. So mother-pup interactions, tactile stimulation. There would be one group extreme with low licking and grooming, and one with high licking and grooming. They also took along arch-back nursing, which means that if the pups have to drink, whether the mother would be like this, and it would be easily reached, or like this, which would be very difficult. But in this case, they look at low licking and grooming, high licking and grooming. And what they could show is that in the extremes, when you look at HPA active responsiveness, then the offspring of the low licking and grooming would have a high HPA active responsiveness. Now the first paper I want to show from this work is a cross-fostering, a switch experiment. So like I said, if you, this terminology of good mothering and bad mothering, by the way, is from a popular science magazine. I don't know whether that's good terminology, but it serves the purpose here. So when you take it, these two extremes and you look at the offspring, what they did is switch experiment. So pups are being born, and then part of these pups are switched to the other side. And what they found is that the foster mother actually determines the phenotype. So if you are born out of a bad mothering situation, but you're transferred into a good mothering situation, then you get a phenotype of the good mothering. So the HPA active responsiveness when you were born out of a bad mother but put into a good mother's nest would be that of the good mother. And the reason I think it's so important is if you look at this spread in the two extremes, then maybe you think, okay, this is just some heterogeneity in this population and there's some polymorphism that will determine what you will find. But this shows that it's really the environment. It's the behavior, in this case probably, of the mother that determines your phenotype. Now, some years ago we actually did the same thing in a maternal separation model. With some interesting results, and I'll walk you through this scheme. We start with a P generation, that's a normal mom and dad, and they get offspring, the F1, and the F1 is subjected to maternal separation, yes or no, so the normal maternal separation model. Left side is a grown-up maternal separation female. When we measure her, adult, give her word avoidance stress, like I showed in Emerald Myers' example, we will see visual hypersensitivity. In this case, she was allowed to mate with a normal male. And they got an F2 offspring, and this F2 offspring was not subjected to maternal separation. And what you see is that the phenotype, so increased response to distension, visual hypersensitivity at adult age, was still passed through to this F2, although we did no longer intervene with this. So the F1 phenotype is passed on to F2 without us intervening. Then what we did was a switch. So we take pups from the maternal separation model, we take pups from the non-handled mothers, and we switch them. And then it turns out we find the same kind of thing as Michael Meany showed, that the enhanced response to stress that you would find in maternal separation offspring, when you put them in a nest of a non-handled mother, you no longer see it. Now, the reason to show this is not because we should all stop looking for polymorphisms and whatever. Please not, but it does show you that the environment is very important, and I think we should never forget that. Environment is very important in the things that we investigate. And that's also the reason that the rest of this presentation will mainly deal with environment-induced changes in HPA axis responsiveness. In this model to start with. So I already gave this away. When you select for high licking and grooming, low licking and grooming offspring, they grow up, you subject them to, in this case, a 20-minute restraint stress, and you measure before and after. What you will see is increased HPA active responsiveness. In this case, they looked at plasma ACTH and corticosterone. And the black ones are the ones with the low licking and grooming, so the bad mothering. Now the question, of course, is how does this happen? And the first thing you think about is the negative feedback of the HPA axis. Is this impaired in these offsprings? Now, a long time ago already, 1990, Paul Plotsky showed that for this negative feedback, of course, you need corticosterone interacting with the glucocorticoid receptor. That glucocorticoid receptor in the hippocampus is essential in this. So what they did was focus on glucocorticoid receptor in the hippocampus. 97 already, a long time ago, they could show that low licking and grooming associated with lower glucocorticoid receptor expression in the hippocampus, and vice versa with a high. So that made sense. But how was it regulated? And for that, they started focusing on the promoter region in the glucocorticoid receptor gene. As it turns out, there's splice variants possible. And in neurons, you have an exon 1-7 that is only present in these neurons. It's neuron-specific, this exon 1-7. And the exon 1-7 is interesting because it has a response element for NGF-IA, nerve growth factor inducible factor A. And if the response element interacts with the transcription factor, you get increased transcription of the glucocorticoid receptor. Now what they could show is that in the high licking and grooming animals, there would be more usage of this particular splice variant. And what they knew already from this other model that I talked about in the beginning, with the short-term handling where you would have a lower HPA axis responsiveness, in that model they already knew that there would be an non-serotonergic tone. And via the 5-HT7 receptor and a cyclic AMP-dependent mechanism, that would give an increased level of NGF-IA. Now putting things together, the increased usage of the exon 1-7 and the increased level of the NGF-A, you can see why we get here, in this case, the increased glucocorticoid receptor expression, and therefore a good negative feedback. Now there was one problem with all of this. When they looked more carefully, they did this in neonates. And when they looked in the adults, there was no such thing as an increased NGF-IA expression. So they went further to find out what was going on in the adult animal. And that's where the epigenetics comes in. And in this case, the methylation. It's known that if you have these CpG islands, and if they are methylated in the promoter region, then usually you will find lower expression of the gene. So they started looking into methylation of CpG islands in, of course, the NGF-I response element. There's two sides of it, 5xn and the 3xn. What they could find in the 3xn, the CpG island over there, if you look in high licking and grooming or low licking and grooming, it doesn't make any difference. In both cases, there will be methylation of this CpG island. On the other hand, if you look at the 5xn end, then in the low licking and grooming, you could find methylation that you would not find in the high licking and grooming, suggesting that methylation at this spot would make the difference for the glucocorticoid receptor expression. I told you in the beginning about the switch experiment. They did it twice, also in this paper. And this is nice because I told you about the phenotype. It would adapt to the foster mother. And in this case, they tried to find out whether that would correlate with this methylation. And indeed, that turned out to be true. So this shows the power of this switch experiment. So I really like those. So that was methylation. Now, if you read into epigenetics, a lot of times you will also read about histone acetylation. You know that the DNA is tightly wrapped around these histones, and that makes transcription kind of difficult. And you get it more accessible by acetylating these histones. So if you need high glucocorticoid receptor expression, you would need increased accessibility of the exon 1-7, and that you can get to facilitate by high histone acetylation. If that happens, you kind of free the NGF-I response element, and you would expect more NGF-I to be attached to it. And those things they looked for, and of course they found those. They did a chip assay, used antibodies to precipitate protein DNA complexes. They had some prior knowledge, anti-acetyl H3K9 histone 3, acetylated histone 3K9, and also NGF-I promoter. And what they found was indeed more acetylated histone 3K9 and also more NGF-I bound to this promoter region. So when you put all this together, then now we have a low licking and grooming and a high licking and grooming. In the low you will find more methylation, and in the high licking and grooming you would find more histone acetylation. That would explain everything in rats. And that is a problem, because how are you going to translate this hippocampal story to a human? Well, they did a good job in that. They took post-mortem hippocampal samples, and they had a control group and two groups that committed suicide. And one was with a history of childhood abuse, and the other was not with that history. They compared the control and the suicide non-abuse. They were never different. You can see that in all three graphs. Left side is total glucocorticoid receptor messenger RNA. And you can see that the total glucocorticoid mRNA is lower in the suicide victims that were abused in childhood. Same goes for the F1 1F splice variant. That's the kind of human homologue of this 1,7 that I kept on talking about in the rat. You see that the usage of this exon is lower in these suicide victims that have been abused. And vice versa when you look at the methylation of the promoter region. This has not been translated to IBS, but it has been translated to IBS-like animal models. And for instance, the maternal separation model that I talked about before. This is very early. This is again Paul Plotsky. Here he compared this handling model, so this lower responsiveness, with maternal separation model, with high responsiveness. And he looked at glucocorticoid receptor mRNA. And you clearly see the difference, and it's very low in the maternal separation model. You also, this paper, this is from a Korean group, and they showed this year, a couple of months ago, in the maternal separation model, looking at the hippocampus, that there's decreases in exon 1,7 glucocorticoid mRNA. Fits with the other model. And they could also show reduced histone 3 acetylation at the promoter 1,7 site. So what about other models? That was predisposed animals. You do something in youth and then they have this phenotype when they're adult. This is another model that's often used in IBS research, and it's adult animals, and 10 days in a row you will give them water avoidance. And it turns out, the left paper is from Marielle LaRouche from LA again. It turns out that these animals, they will have an increased HPA axis responsiveness and will have visceral hypersensitivity, so it fits like an IBS model. And what you could show is that it's really HPA axis dependent. When you, again, blocked central CRF1 receptors, the model was gone. Now this model was used by the Wiley group in Ann Arbor. And in this case, they didn't look at the central mechanisms, but at peripheral mechanisms. So DRG neurons, the afferents that are important in visceral hypersensitivity. And what they showed is hypermethylation in the same exon 1 region, exon 1,7 region that we talked about before, the promoter region in these DRG neurons. And that correlated with lower glucocorticoid receptor expression. For what? What does it mean? Well, they found some nice extra evidence what it would mean, and that is lower CB1 expression, lower expression of the cannabinoid receptor 1. It turned out that this promoter of the CB1 receptor has a response element for the glucocorticoid receptor, and evidently if the glucocorticoid receptor is low, you would find lower CB1. And also they found increased methylation in this CB1 receptor promoter region. And all of that led to the conclusion that this would have a negative effect on endogenous analgesic mechanisms if your cannabinoids can no longer function because the CB1 is not there. You don't have the analgesic mechanism. This was a very interesting paper because not only they showed this, but they also looked at TRPV1. And they could show that TRPV1 is, the expression of TRPV1 in these DHG neurons would be increased. The TRPV1 is a ligand-gated ion channel, which is very important in pain sensation, also in visceral pain. So what they show here in this model, so 10 days water avoidance, the baseline, what is that kind of color? Bluish. The lower line is control animals. The orange, I think, the upper line, this line is after 10 days water avoidance. You get an increased response to distension. This is visceral motor response again. If intrathecally during this 10 days water avoidance, they give intrathecal TRPV1 siRNA, you see that the response is completely gone. What you also see is this one, EP300 siRNA. Now EP300 is a HAT, histone acetyltransferase. What they could show is that this HAT is bound to the TRPV1 promoter, that this correlates with the increased TRPV1 expression, and it could also show that if you block the glucocorticoid receptor with a pharmacon, that you lack the upregulation of this EP300. So really there's different steps to show that indeed TRPV1 is perhaps a stress-responsive gene. Well, I had some problem when I read this because I thought, well, this is all very nice, upregulation, and that's why they have this pain. We also looked at TRPV1 in the maternal separation model, and we found an important role for it. If we had a baseline response, we gave stress, it would be increased. With a selective antagonist, it would be reversed. But there was no increased expression. And when Guy Boekstaans and Mira Bouters in Leuven did patient studies with this abastine, the histamine 1 receptor antagonist, they also did some experiments with TRPV1, and they could show that in the absence of upregulation, there was an important role for TRPV1 in all of this, because activation of the histamine 1 receptor would sensitize TRPV1. Sensitization, not upregulation. So how can you correlate these papers? Well, in the discussion of the upper left paper, they kind of gave it away. I think that's really, well, it's my opinion that is true, probably. There is a dual role for endocannabinoids. Anandamide, endogenous cannabinoid, can directly activate TRPV1. You get a pain response. But of course, it can also activate CB1, and then you get an analgesic effect, because then you can inhibit the phosphorylation of TRPV1. And probably that's what's going on. It showed in this model downregulation of CB1. And obviously, if you don't have the receptor, then perhaps you go to the left side of this picture and you get pain response. It's just an explanation that I think may be true. Now, for the last part of this presentation, I'm going to try and convince you that perhaps there's a connection between this stress-responsive gene TRPV1 and something that happens in this newly discovered stress-responsive organ that we have, which is, of course, the microbiome. I'm not really going to talk about the microbiome without a mycobiome. Mycobiome is fungi in the gut, and it's very small, the microbiome. It's less than 1, perhaps even less than 0.1%. We don't really know what the numbers are, but it's a very low part of the microbiome. But still, it can be very important, at least in this model. What we could show is that the gut microbiome is indeed responsive to early life stress. If we compare the microbiome, we did ITS1 sequencing, and you look at maternal separated animals and non-adult animals, there's a huge difference. There's a really different microbiome in these animals. On top of that, if we use these maternal separation animals, we give a stressor, increased sensitivity to this tension, and then treat with fungicides, we can reverse this increased physical hypersensitivity. And also we did some transfer experiments, fecal transfer, to show that the difference that you see between maternal separation and non-adult is also relevant. But I didn't put the result here. Now, on top of that, what we could show, what is relevant here, is really the recognition of the fungi or fungal antigens by the immune system. If we blocked recognition of fungal antigens by blocking dectin-1 signaling or with soluble beta-glucans, this dectin-1 recognizes beta-glucans that are in the fungal cell wall, or we inhibit the signaling, which is via SIG, then we lose the response. So recognition of fungal antigens is relevant. And what you would also show in vitro is that if you incubate your mast cells, in this case HMB1 cellan, if you incubate them with fungal antigens, you get histamine release. One small step back, and then you understand why I'm telling you all of this. In this maternal separation, we showed an important role for TRPV1. Antagonists could reverse the existing hypersensitivity. We could also show the relevance of the histamine 1 receptor. Avastin treatment, histamine 1 receptor antagonist, could reverse existing visceral hypersensitivity. And Guy and Mira in Leuven, they showed in patient biopsies that when you occupy the histamine 1 receptor, you will get the sensitization of TRPV1 and therefore your pain response. Now, what was lacking in this picture is how is it being activated? Why do you get histamine release? And, well, I don't know the answer for patient, but for this model anyhow, we could show that this is perhaps fungal antigens. So an interaction of mast cells with fungal antigens would give you the histamine and therefore an important role for TRPV1. And I think this is the last slide. So I talked a lot about genes and epigenetic modulation of genes in the HPA axis and in relation to that visceral hypersensitivity. I did not talk a lot about a lot of other things, sorry for that. Your favorite molecule pathway next time, that will be the topic probably. So thank you.